Should animals be used for biomedical research? Before we answer that question, we have to look into why scientists use animals for biomedical research. Animals like yeast, fruit flies, mice, rats, frogs, and chick embryos all have a close phylogenetic relationship to humans, which provides similarities in genetics, behavioral, and biochemical activities. Because of this, animal research has been an important contributor, adding 25 years of life expectancy from the start of 1900s to present day. But despite this, despite the many advances, some people question the accuracy of animal models to study human disease. This leads me to my plan, which is yes, we should continue the use of animal testing for biomedical research for now. However, it can be reduced, and we can transition into animal-free research. First, we look at animal models, which in a study by Professor Rits Huytegen from Radboud University in the Netherlands, it was found that 90% of the potential medical interventions from animal models failed to repeat the same in human clinical trials. Furthermore, in a study by Pharma, 2/3 of the repeated studies gave inconsistent results with earlier published results, leading to a discontinuation of these projects. These unrepeatable projects of animal research has estimated to cost $28 billion annually in the U.S. alone. One such solution to combat the poor transition of animal testing to human clinical trials is systematic reviews. Systematic reviews criticize past experience involving animal research on a certain topic, which interprets the strength of the design, the methodology, and results of it. This would help create a more accurate model from animal models to humans, which would ultimately determine the safety and efficacy of proceeding with the trial. This again would lead to a decrease in animal use and a better design, quality, and reportings of these experiments. An example of this is at Radboud University, which decreased their animal use by 35% by using systematic reviews. Systematic reviews also lays the foundation for better research and better outcomes for the future of biomedical advances. This leads me into my next point, which is using organ-on-chips models as an alternative to animal models. Organ-on-chips are microfluidic culture devices that replicate the complex structures and functions of living human organs. The presence of fluid flowing in the model allows it to mimic both the cell experiences in the body, such as how it receives nutrients and moves waste, and how the drug will move in the blood and interact with multiple types of cells. The ability to control the fluid also allows researchers to fine-tune the optimal dosing for a particular drug. However, most models are single-use and allow only one input. This can limit what research can study at a given time, as well as they are both expensive and labor-intensive to work. Further, the significant upfront cost associated with using the model could potentially discourage its widespread use for biomedical advances. In conclusion, animal use is a necessity until technology becomes more advanced and we can reduce the amount of animal use using the processes like systematic review.
Thank you. What information did you need you weren't able to find, and how did you go about trying to find that information?
One thing I couldn't find was along with the Radboud study about how they reduced the animal use by 35%. I wanted to see if along with like the decrease of animal use, it did improve their experiments, but I couldn't find it on like their website, their biomedical page.
Okay, thank you. How does your conclusion respond to any other research or sources you examined?
My conclusion responds to one college page I found, which was like, which had a strong opinion about like why animals should not be used. Like it kind of pointed like an ethical standpoint of it, and like it responded saying that like it's like kind of we need it for now until like things like I guess the organ chip comes to play. But yeah.
Okay. Thank you. Another round of applause.